Thank you so much, Dr. Johnson. Great talk. And rounding us out here, Dr. Joe Bellucci from Rarebase. Title of his talk is Applying a Precision Medicine Drug Discovery Platform to SURF1 LS Syndrome.
Okay, thanks, Ethan. Hopefully you all can see my slides and a picture of our lab at Rarebase in Palo Alto.
Looks like it's still loading, perhaps.
Give it a second. Let me try it again. It says it's paused, so.
Yeah, it looks like the same thing happened 2 talks ago. I'm not sure how that was corrected, but maybe. Let's see.
Yeah, still saying pause, which I've never seen before.
Can you try it again now?
Still showing paused.
Yeah, not sure what the AV issue is here, unless you're prepared to do a whiteboarding session here. Maybe logging in and out. I don't know if it's one of those things, or
Try one
maybe
more.
it's if you have a split screen, or it's.
Try one more thing.
Joe, did you put your talk into Dropbox?
There it is. We see it now.
Working now. Yeah. Okay. Okay. So now hopefully you can see a picture of our lab. But yeah, thank you to the organizers for inviting me to speak today. First of all the talks have been amazingly interesting, and we're very excited to share some of the work that we've been doing at Rarebase as it applies to SURF1 LS syndrome and our drug discovery platform for precision medicine. So I wanted to talk a little bit first about Rarebase's general approach, and then move into how this approach is being applied to LS syndrome and SURF1 specifically. So we're taking really a multi-pronged approach to identify drug candidates for rare diseases. And we're primarily focused on trying to repurpose all of the 2,700 FDA-approved drugs across rare diseases, not just LS syndrome, but across the full spectrum of genetically defined rare disorders. To do that, we're developing data sets in-house, platforms, and facilities to support this. And the hope is that by looking at the connections between rare diseases, that we can find unexpected things, increase the efficiency of research, and ultimately bring effective therapeutics to patients across disorders. And Rarebase really, I think, provides a path to move drugs to patients as quickly as possible in an efficient way, and really in a way that generates decision quality data that can be applied to the clinic. So what we're doing from a process standpoint is to take patient-derived or genetically engineered cells that have the background and the right context for studying a disorder. And then we're looking in our in-house database, which we call Function. And this is really a database of drug screening data and a variety of other data sources that we're generating that enables us to computationally predict candidate drugs that may be effective for a specific disorder or a specific genetic variant. And that's where the disease context of the patient-derived or the engineered cells becomes really important, because we take those cells and test the drug predictions in those cellular models of disease to generate, like I said, decision quality data that can be used in a potential clinical application. And really an important thing to emphasize, and I think this is what motivates all of us at Rarebase, is that our discovery research approach begins and ends with the patient in mind. Our goal is to find effective treatments for people, and we're aiming to do that in a way that's efficient, fast, thorough, and collaborative. So we're very open to working with not only other companies, but other research groups, as well as individual families and patient advocacy organizations to get to this ultimate goal of bringing effective therapies to patients. And just to go into a little bit more detail about our drug prediction database, if you will, which we're calling Function. This is really the engine for our entire operation, and it rests and relies on three main pillars. The first is insights from those disease-relevant cell models that allows us to really measure the underlying biology in a very thorough and disease-specific way using a variety of experimental methods. A second pillar, as it relates to one of those experimental methods, is insights that we're gaining from drug screens. And this is what I'll talk about in a lot of detail in the following slides. But we're generating really rich data sets across the druggable genome and using that to identify novel opportunities for drug repurposing and for novel drugs. And then finally, the third pillar of Function is really insights from computational analyses. So many of the methods that we're using are generating really large data sets that require kind of next generation computational techniques to both aggregate the data and then to synthesize it in order to make those drug predictions as efficient as possible. And the combination of all these different sources of data and techniques is really what we think will maximize our ability to find effective drugs. So on the drug screening side, because this is kind of the main experimental approach that we leverage, this is really producing a set of rich transcriptomic data that enables our drug candidate prioritization. The process for the drug screening that we're doing is shown in this bubble, in this flow chart. Starting at the left side, we take iPSCs, which you've heard a lot about from previous speakers, and differentiate those into neurons. And then we treat them with a compound library. So our initial library is about 4,000 different compounds. Many of those are FDA approved. And the neurons that we're using for the screen here are derived from an iPSC line that comes from a presumably healthy volunteer. So there's no sort of disorder specificity here. We're testing these drugs on essentially healthy human neurons. We do the drug testing all in parallel. So it uses these plates that many people on the call may not be familiar with, but they're 3D4 well plates where each well is getting a specific drug, and we're treating neurons in that well with that drug. Each of these wells is about the size of the tip of a pencil, like a pencil lead. So very small, and we're able to run this with automation in a very high throughput way to be able to screen a lot of compounds in parallel. One differentiating feature of our approach is that we're not just interested in a specific gene's readout, but we're actually measuring essentially all genes in these cells and how they change in response to each drug. And we're using a technique to do this called 3'N pooled RNA-seq. And essentially, this is where kind of the power of our approach comes into play, because we can take the data that's generated and query it for a specific gene and start to identify which compounds either increase or decrease the expression of that specific gene. And we can do that across essentially all genes that can be measured in these cells. And we're reliably able to measure around 10,000 different genes at this point in these cells. The data that this is generating is really massive, I should point out. Compressed so far from this library, the compressed data set is around 30 terabytes. And just to put that in perspective, if you were to move all that data onto, audio CDs, it would be about 50,000 audio CDs just from this initial set of 4,000 compounds that we've screened. So that's where the computational analysis of this data becomes really important, because we're really dealing with truly big data sets. So I'd like to share our preliminary results for SURF1. And I'll talk about this plot in a little bit of detail in a second, but I think it's important to focus on the key takeaways first. So of those 4,000 drugs that I keep mentioning, we've screened about 2,800 of them so far, and this includes a large number of FDA-approved drugs. We're also in the process of sort of finishing out that set and have data coming in on 1,200 additional compounds, which are not shown on the plot at left. But we are identifying preliminary candidates that are able to change SURF1's gene expression. An important caveat here is that each drug in the screen has only been tested at a single concentration, and the effects of any of these candidates need to be confirmed in follow-up studies, which we're planning to do. An important feature of that is to look at a very thorough dose response characterization of each of the candidate compounds in a patient-derived cell line rather than in healthy neurons or in PCs. And so the goal here with SURF1, I also want to point out, is to be able to boost the expression of copies of SURF1 that have missense mutations that have presumably some residual activity. This strategy clearly won't work for cases where there are truncating mutations in both copies of the gene. That would essentially be like if you have a car that has a flat tire, we can put air into it, and that's sort of the missense mutation case, and the car can drive more efficiently. In the truncation case, where both copies are broken, that would be like a car with no wheel. Putting air into it isn't going to make much of a difference. So it's really for this specific use case. I should point that out. The plot is showing our initial set of results. The horizontal or x-axis is showing the fold change in SURF1 gene expression, and each point on the plot represents the response of SURF1 expression to a specific drug or chemical compound. The FDA-approved drugs are highlighted in green. Non-FDA-approved drugs, these are generally clinical stage candidates, but there are some preclinical candidates as well. Those are highlighted in pink. And we are also looking for whether any of these compounds negatively affect cell viability, and those are denoted in gray, and those would be sort of excluded from further follow-up because they're essentially toxic to these neurons. The y-axis for the vertical is showing how confident we are in each of these results. This is the minus log 10 of the p-value for those that are more technically focused. So essentially being higher on the vertical axis indicates that we have more confidence in the ability of a specific drug to move SURF1's gene expression up. And so we're very excited about these results and are planning, follow-up studies on things that are increasing SURF1, moving it to the right in this plot, and also that we have good confidence in. And I do want to present some interesting sort of aggregated analysis that we're starting to do on this data, where we've identified two potential classes of therapeutics that may be able to increase SURF1's expression. So the first class, target class A here, is highlighted in pink. There's actually 7 different molecules that are all of the same class that we've identified in these results that are able to significantly increase SURF1's expression. 3 of those are FDA-approved drugs and 4 are clinical stage. So this is really exciting because this is essentially reinforcing that this target may be valid as a therapeutic option. There is also another class, target class B, which is highlighted in green. One of these compounds is a tool compound where it has known toxicity, so it's not actually being clinically developed, but the other 2 are clinical stage drugs. And just to focus on that target class B here in this bubble on the top right, this is sort of trying to show an exciting feature of these 3 drug candidates. One of them is a known activator of a protein that leads to SURF1's gene's expression increasing. The other 2 candidates are actually inhibitors of an inhibitor of that SURF1 activator. So we're seeing candidates that are popping up across different protein targets of the same pathway. And we're also seeing for target class A compounds that are hitting the same target So this is really exciting across both of these target classes. We're obviously hoping to move these forward into additional testing. And that kind of leads to the key takeaway, which is that we're seeing multiple candidate molecules with these overlapping mechanisms of action. And this is, motivating our additional characterization and development efforts. So future directions for this program are that we've generated some patient-derived iPSCs. We're hoping to generate more in the future to be able to test these drug candidates across a variety of genetic backgrounds that are specific to multiple patients. We're in the process of generating neural progenitor cells and neurons, depending on how easily those can be differentiated, which, previous talks have discussed the challenges of doing that, but we're hoping to at least get to the NPC stage and then to test these candidate drugs in these confirmatory studies that would look at not only SURF1's gene expression, but also start to assess functional improvement in neurons or NPCs via a high content microscopy, which we have up and running in our facility, as well as gene-specific functional assays like mitochondrial fitness assays. Ultimately, we hope that this panel of data can be summarized for physicians and used for potential clinical application through either repurposing of FDA-approved drugs or continued development for drugs that are not FDA-approved. And just to sort of highlight one of the functional assays that we're hoping to use and that we've stood up through a partner that we're working with, we are looking at mitochondrial oxygen consumption in SURF1 affected cells. This is sort of a standard assay that many people may be familiar with that runs on Agilent's Seahorse platform. And this essentially measures how healthy the mitochondria of cells are in response to various stressors. And I think the key takeaway, to not really belabor this in the interest of time, is that we've stood this assay up with SURF1 patient-derived fibroblasts and look at and have been able to look at an unaffected control relative to those cells, and do see a significant difference in their basal level of respiration between the two cell types. So this is the type of thing that we're thinking about as a corollary to our drug screening efforts that are really focused on gene expression and microscopy. The functional assays, I think, are a key additional layer to that hopefully inspires clinical translatability of these compounds going forward. So with that, I'd like to thank not only the Cure Mito Foundation and the organizers of this amazing conference, but also the entire team at Rarebase. We're about 25 in Palo Alto, and it's a great cross-functional team. Everyone has contributed to this work and the work that's being done across the variety of rare diseases that we're working on. So with that, I'll end, and happy to take any questions in the follow-up session.
Great, thank you so much. I think we're